JAK STAT signaling pathway. Components of erythropoietin-mediated JAK STAT signaling pathway. The first component is erythropoietin. It is abbreviated as EPO. Structurally, it is a peptide. It consists of 165 number of amino acids. It is a cytokine or developmental signal. It is produced inside the kidney and it increases the erythrocyte production or RBC production. There are more than 30 cytokines and hormones that activate JAK STAT pathway by binding to cytokine receptors. For examples, growth hormone, prolactin, leptin, GM-CSF, alpha interferon, gamma interferon, etc. Thereafter, the second component is the erythropoietin receptor monomer, or abbreviated as EPOR monomer. It is a type of cytokine receptor. It is present on the plasma membrane of a cell, and this is the extracellular space which is present outside the cell, and this is the cytosol which is present inside the cell. The part of the EPOR monomer which is present at the extracellular space is known as extracellular domain, and the part of the EPOR monomer which is present inside the cytosol is known as intracellular domain. Now the extra facts: cytokine receptors are dimer or trimers, means they may contain 2 or 3 distinct bodies. Then the third component of erythropoietin mediated JAK STAT signaling pathway is the JAK2 or Janus kinase 2. It is named after the two-faced Roman god Janus. It is a type of tyrosine kinase enzyme and it is water soluble in nature. It is present in the cytosol of the cell, and other known JAKs are JAK1, JAK3 and TYK2. TYK2 stands for tyrosine kinase 2. Then the next component of the JAK STAT signaling pathway is the STAT5 or signal transducer and activators of transcription 5. It is a gene regulatory protein which switch on or off any specific gene present in the DNA, or it acts like a transcription factor. It is present in the cytosol. There are 6 starts found in mammals. The STAT5 protein generally contains an SRC homology domain, which is abbreviated as SH2 domain, and SRC is a soluble protein tyrosine kinase. It is associated with certain receptors when they bind to their ligand. SRC was the first protein in which we had found phosphorylated tyrosine binding domain. This phosphorylated binding domain subsequently named the SH2 domain. Now, the signaling pathway. Whenever there is low level of oxygen in the blood circulation, which is known as hypoxic condition, the kidney produces a hormone known as erythropoietin. This erythropoietin is delivered to the extracellular space via the circulation, and the erythropoietin generally binds to the extracellular domain of EPOR, and after binding to the erythropoietin or EPO, the EPOR get dimerized. These are the JAK2 proteins. The JAK2 generally binds to the intracellular domain of EPOR, and after binding to it, the EPOR can phosphorylate each of the JAK2, and after phosphorylation the JAK2R get activated. Now these activated JAK2 can phosphorylate at several tyrosine residue present in the each EPOR monomer. Now another protein or known as transcription factor STAT5, which is present in the cytosol of the cell, now move towards this phosphorylated tyrosine in the EPOR monomer, and the phosphate group binds to the SH2 domain of STAT5. After binding to it, the phosphorylated STAT5 molecules or activated STAT5 molecules are get detached from the EPOR. Now they are released to the cytosol of the cell, and now the phosphorylated STAT5 molecules move towards each other, and phosphate group of each STAT5 molecule will now bind to the SH2 domain of each STAT5 molecule, and this binding will form a homodimer. Each each of STAT5 molecule generally contains an NLS domain or nuclear localization sequence domain. This drive the homodimer towards the target DNA present in the nucleus. Now the homodimer, STAT5 homodimer, will move into the nucleus through the nuclear pore or translocated into the nucleus through nuclear pore. Now this STAT5 homodimer will bind to specific DNA sequence present in a DNA and activate it. STAT5 homodimer along with other protein will activate the specific gene responsible for the erythrocyte production, and now the transcription will occur, and after transcription mRNAs are produced or messenger RNA are produced, and these mRNA will form different proteins to produce a physiological effect. Thank you.